Thank you all for joining us as a good group, and we could have similar group in all the adult education in the future. That'll be great. I want to take a minute to welcome somebody we all know well, Rachel Presti. Dr. Presti is with the Washington University. She is the Associate Professor of Medicine and also the Medical Director of the Infectious Disease Clinical Research Unit, as well as the Principal Investigator of the NIH AIDS clinical trials at WashU. So she got her MD and PhD at WashU, and her clinical and research interests and her publications cover a wide spectrum in the field of infectious diseases, including improving the care of HIV-infected patients who are co-infected with emerging viruses. But many of us have seen Dr. Presti during the past year on the television and on other news reports, answering questions about COVID-19 and the development and rollout of the vaccine. So we were all really wanting to hear what's going on. And first of all, what's this virus all about? And what's happening with the rollout of the vaccine, and what can we expect? So we are really blessed to have a scientist with deep knowledge in this subject among us. And I really want to welcome Rachel to us. And for those of us who may not know, Rachel is a member. Rachel and her family are members of Trinity, longtime members of Trinity. And so it's a great pleasure for us to have her with us. So she will start with a short presentation, and then we have time for Q&A. To do the Q&A, we have collected a few questions from members of Trinity, and I have passed that on to Rachel. And after that, we can answer other questions. You can, I think it may be a little hard if you raise your hand, but you can also enter in the chat box, and we will be monitoring that. So without further ado, Rachel.
Well, thank you very much. This is a nicer audience than I sometimes get. So it's nice to talk to everybody. So this is sort of a, let me see if I can make this work properly. Can people see the slides? Is it the sharing working?
Yes, it is working.
Okay, all right. So yeah, so this is taken from a presentation, or the presentations I've done for community folks. And so it's just a couple of slides, but I figured it gets everyone kind of on the same page as to what we're talking about. And so just to start, this is, I know it looks complicated in sciencey, but this is sort of a primer on the immune system. And mainly the point I want to make is that the immune system is a lot more complicated than just antibodies, even though that's what we talk about a lot. And so this is a model. I mean, it's RSV, which is a different virus, but another respiratory virus, very much like coronaviruses. So it infects, and actually your initial immune response is not very specific. It's directed against any sort of invasion or danger sort of signal that the immune system detects. And you get a lot of inflammatory sort of proteins that frankly are the same kinds of things that make you feel badly. So it's those proteins that make you feel icky when you get the flu or a cold. And then you have these sort of innate immune cells that initially kind of limit the damage but can't clear the infection. And then you get this acquired immunity that you know, we always talk about antibodies, and that's really, really important. Antibodies actually prevent the virus later from coming in and infecting cells, period. But if cells do get infected, then there are T cell responses as well that are to different parts of the virus. And so that's to get us kind of on the same page. This is just showing just that adaptive immune response, that specific immune response against a pathogen. And so we talk a lot about this, the B cell response and the antibody response, but there's a T cell response as well. So what we are able to measure easily clinically is the antibody response. But you should know that your body is much more complicated in terms of how the immune system actually focuses on a pathogen and has a specific immune response, so that if you get vaccinated or if you get infected, the next time you see that, your immune response is much, much faster and really prevents you from getting sick the next time you see that. And that's sort of how a vaccine works. So the first time you see an infection, that's shown there kind of on the left, you make these antibodies. They're not actually really great antibodies initially, and then your body trains on the pathogen to figure out, okay, these are the initial antibodies that work, and it makes them more specific over time. So your immune response does start to occur about 7 days after exposure to a vaccine or a pathogen. You start to get that specific immune response. It tends to actually improve over time. And then the second time you're exposed, you have a much more robust kind of targeted response. So vaccines work basically by classically by introducing a weak or an active form of the infection, the virus or the bacteria or other pathogen. What we're doing more recently is, as we know more about these infections, what we're actually doing is figuring out what part of the virus is important. And so for the coronaviruses, we figure that the spike protein on the outside of the virus is the most important thing to make an immune response to. That seems to be protective if you have an immune response to the spike protein. And so then you don't have to introduce the whole pathogen, and you don't have to introduce a weak pathogen that might be problematic for people whose immune system is not very strong, or might cause more disease or side effects. You can just introduce the part of the virus that you need to make an immune response to. And then your body makes those antibodies and also a T cell response, and then they remember it the next time. And so if you're exposed a second time, you have a good immune response, not always preventing the exposure or the infection, but preventing you from getting sick from it. So the vaccines that are currently available, Moderna and Pfizer, are mRNA vaccines, which is a new kind of vaccine. So there are no currently available mRNA vaccines for other infections, but that doesn't mean the technology is new. So people have been working on RNA vaccines for at least 10 years. So there's lots of experience and lots of trials with RNA vaccines. This just happened to be the first one that really moved forward and clearly worked very, very well. And so basically it's a really clean and kind of nice way of doing a vaccine. You take the spike protein RNA sequence, which is sort of the blueprint that your cells need to make the spike protein. You wrap it up in a little lipid coating so it gets into your cells, and then you inject it. It gets those little lipid balls with the mRNA inside, get into your cells, make the spike protein. They also happen to induce the sort of danger signal that your immune system needs to say, this is something I should make an immune response to. And then you make the antibodies against the spike protein. You also make fairly decent T cell responses against the spike protein as well. So a couple of things to remember: that the immune response induced by the vaccines is just to the spike protein, not to the entire coronavirus. And so that becomes important a little bit later. But the other thing is mRNA never gets incorporated into your DNA or your cells. It gets in, it makes the protein, and it gets very rapidly degraded. And that rapid degradation is the reason that those vaccines need that really cold storage, because they don't last very long. once you thaw them, because there are ways of degrading that rapidly, which is nice. It means that it's not sticking around and causing side effects or other harm, but it does mean you need that cold storage. So this is kind of the advantages of those RNA vaccines. The new vaccines that are coming out from Janssen and AstraZeneca, which are expected to be, at least Janssen is at the FDA and is expected to be approved by the end of this month. Those are viral vectored vaccines. So there are approved vaccines. The Ebola vaccine is a viral vector vaccine. There's a lot of work that's been done on these. Even though they're called viral vectors, they're basically sort of weakened cold viruses, adenoviruses. They can't replicate, they can't cause a cold. So their main goal is then to get the DNA that you need to make the spike protein into the cell. And then they also don't stick around a long time. And they do a good job of stimulating the innate immune response and the T and B cell response, the same as RNA vaccines do. So you get good immunity, and none of them sort of integrate into the cell. They're sort of naturally degraded. They stimulate the immune response pretty quickly. The advantage of the viral vector vaccines is that you don't need cold storage. So they can be in the refrigerator for 3 months, which makes them much easier to distribute. And we'll talk about some of those in just a little bit. So this is the spike protein. So if you look on the left, that cartoon has is of the coronavirus, and those blue knobby looking things sticking out, that's the spike protein. So if you think of the virus as like a ball, all around the outside of it is studded these little spike proteins. And so you could see that if you had an antibody that could bind to those, you would prevent the virus from getting into your cells and causing an infection in the first place. One of the important parts of that spike protein is shown in the sort of blown up version there as that purple piece. That's the receptor binding domain. That's the part of the spike protein that binds to a receptor on your cells called ACE2 and allows the virus to get into cells. So that's clearly an important part of the virus. And if you make antibodies against that, you could see how that would prevent your the virus from getting into cells. And then there on the right is just one of the early animal studies with one of the vaccines that was directed against the spike protein. And you can see if you use a sham vaccine, so a vaccine that doesn't have the spike protein, all those black wiggly lines is all the virus that's replicating in those animals. And if you vaccinated them so this was using multiple different sort of slight variants of the spike protein. And this was one of the really good ones where you saw no replication then of the virus after you vaccinated with the spike protein. So just some of the animal data. There's all sorts of talk about how we maybe did this way too fast, but all of the steps were gone through, including all of the animal models to demonstrate that this was an effective way of vaccinating. So there is a little bit of a difference, and I alluded to it before, between your vaccine response and the antibody response to infection as a whole, because the vaccine is just directed at the spike protein. But you can see in the little cartoon of the virus that there are all sorts of other things there. There's all sorts of other colors, including matrix protein. There's that red is the nucleoprotein, as well as RNA of the virus. So the antibody tests that people can get commercially were actually made against the nucleoprotein. So if you go and get your antibodies checked, if you go to an urgent care or you go to your doctor and you say, I want to know if I have antibodies against the coronavirus, what they're testing is whether or not you have antibodies against the nucleoprotein, mostly. Some of the tests are against the spike protein, but most are directed against the nucleoprotein, which means that if you got vaccinated, your test will be negative. Your antibody test will be negative. So, so it doesn't make sense to go and get your antibodies checked after you've been vaccinated, because we're not measuring the same thing. And you'll just make yourself nervous that you didn't get a good immune response to the vaccine when you really just got the wrong test done. So it's something that we're hearing a lot from people who are worried about making sure they're immune, thinking, well, I'll go check my levels of my antibodies. And you should be aware that the spike protein that you get vaccinated against isn't the same antibody test. So so you shouldn't do it unless you're worried that you may have been infected, and you want to see if you've had a past infection. Frankly, I'm not sure that there's a whole lot of utility of doing it at this point, period, except in large scale studies to see how many people have been infected in a population. So the other thing to know is, you know, a vaccine induces your body to have an antibody response, an immune response. But we also have these antibodies that can be given as therapy. So, so basically, they're sort of passively transferring an immune response to somebody who's currently sick, or in some situations, who's been exposed to the virus. And so those have some limited efficacy. If you give them to people at the right time, they can prevent people from getting sicker. If you are already making antibodies yourself, they're not additionally helpful. But it is a treatment that is now becoming more available. I think the concern we have is that those antibodies were directed against the original coronavirus, and it's not clear how well they're going to continue to work if we get more variants that are circulating in the community. So, but that is something that you could ask your physicians about if, despite all of your efforts to keep from getting the coronavirus, if you did get sick, if you did test positive, and you're not so sick that you need to be in the hospital, that is a treatment that is becoming available throughout the community. So, okay, so but back to vaccines. This was sort of back, sort of early summer, what the time frame was for the different vaccines that we were looking at. And so the initial vaccines that were going to be tested were Pfizer and Moderna. It's important to know that part of the reason that those were the first vaccines that came up is mainly because they're actually easy to make. So as soon as we knew the sequence of the virus, people could look at what the sequence of the spike protein was, make that RNA, package it into those little lipid balls, and they were ready to go very, very quickly. The viral vector vaccines take a little bit longer because you have to actually grow those in cells, because they are similar to viruses, so they need to be replicated in cells. You can't just make them easily in the lab. And then the vaccines that are probably going to come up a little bit later are recombinant protein vaccines. So these are vaccines where you make the spike protein itself. So the body isn't making it, but you're making it in a lab and then injecting it. So it's a lot harder to make proteins than it is to make RNA or the DNA that you make for a vector vaccine. And so those have been a little slower to come along. But then, and then there are vaccines that are not as talked about as much in the US, but one of the Chinese vaccines is an inactivated entire coronavirus, kind of like the traditional vaccines that we used to give. I mean, we still give them, but that we used to develop, where you just inactivate the virus, you kill the virus and give the whole virus. The time frame for vaccines was the same as it is for any other vaccine or actually any other drug. You initially do, you develop the vaccine, you test it in animals, you certify that it is safe and effective in animal models, and that it is, that doesn't have off-target effects. And then you do clinical trials, and there's really 3 phases to those clinical trials. Phase 1 is usually small, maybe a couple hundred people study, where we look to see in generally in young, healthy people, are these safe? And maybe do we get an immune response? So a little bit of safety, and then a little bit of, what is the dose that works best to get an antibody response. Phase 2 is more dose responsiveness. And so more, a larger study that's maybe got a couple hundred people up to a couple thousand maybe, where you look to see which vaccine gives you the best sort of antibody response in a population that is a little bit broader. So not just young, healthy, but a more diverse population. And then phase 3 are the studies that we do to really make sure that something is effective. So we're still collecting data, we're still collecting information about how you induce that antibody response. But the main point of the phase 3 studies is, does it prevent people from getting sick from the vaccine or from the virus after you get vaccinated? And so those are large, sort of tens of thousands of people studies. And then after that, it goes to the FDA. All the data goes to the FDA. They take a look at it. And the way the FDA worked for this particular, for coronaviruses, was that they put out information about how to do emergency use authorization because we're under a medical emergency. And so all of these are sort of indicated as being stepwise, and you wait between them, but there is a little bit of overlap. So the FDA is involved in the design of the studies. They are looking at the data all along. And for many of these vaccines, then that large scale production and distribution actually started way at the very beginning. And so, so these companies were making lots and lots of vaccines even before they knew whether or not they would work. And the government said, we will buy them if they turn out that they work and they're safe. And so there are a couple reasons that we could go fast, but all the same steps were in place. So these are the vaccines that are currently under emergency use authorization, the Pfizer and the Moderna vaccines, and some information. So basic information about the vaccines. And then on the side there, I have for comparison some of the other vaccines that people get all the time and the studies that were done to approve those. So there were 43,000 people enrolled in the Pfizer study that went to the FDA to get approval. 20,000 of them were over age 56. I'm not sure why they chose that age group, but just so that you know, these are not only tested in young, healthy people, they were tested in older people as well. Pfizer actually did also do their studies in kids, and they've extended that as well now to see if these vaccines work in kids. And then about a third were non-Caucasian, so persons of color, Hispanic, Black, Native populations, Asian populations, so non-Caucasian populations. There was a lot of talk initially that the vaccine studies were not enrolling enough diverse populations. So I'm just putting out the numbers there that was recognized early. And I don't think there was as much publicity about the fact that it was actually addressed, and there was a huge diversity, actually much better than a lot of other clinical trials. Similar numbers were seen for Moderna, and then you can see that 95% efficacy. I'll talk a little bit about that. Those numbers, I think, are very confusing to people because they're not really written in stone. So it's efficacy based on the time period that you're looking, the number of cases, and basically how many cases were there in people who got the placebo vaccine how many cases were there in people who got the vaccine. So you can see there are 162 cases in the placebo and 8 cases in the vaccine, suggesting you prevented 95% of the cases. If it were 0% effective, you would see 162 cases in the vaccine as well, and you only saw 8. So you prevented that many cases. That's where you get the 95% efficacy. And you can see that there were 9 severe cases in the placebo arm and 1 severe case in the vaccine arm for Pfizer. And then the numbers are there for Moderna as well, very, very similar. A little bit more cases in terms of number, more severe cases in the placebo arm there, and no severe cases in the vaccine arm in the Moderna vaccine. AstraZeneca, I don't have all the data. They've done multiple studies worldwide, but they are still, they still haven't published their data from the US run study. That is a 30,000 participant study. And so we're waiting to see what that data looks like. Janssen just put out their data. They enrolled 44,000 people. A third of them were over the age of 60. About 59% were white, 45% Hispanic or Latinx, 19% Black, and 9% Native American. So nice broad population. This was enrolled in the US, South Africa, and South America. The thing that's different, so the effectiveness on that one, it came out as 66% against infection and against symptomatic infection. The difference is that they had 468 cases, and that's mainly because Pfizer and Moderna were enrolling their studies earlier in the summer, and so their cases were coming there in sort of September, October, November. And then Janssen was a little later getting started, and their cases enrolled sort of November, December, January, when we were in the middle of this big spike and we were starting to see variants. One of the things to note is that 57% efficacy in South Africa, 90% of the cases seen in South Africa were the variant virus. So maybe a little bit less effective at preventing infection. The other thing that's important to note, though, is that in the vaccine arm, I don't have all the numbers yet, but in the vaccine arm, they saw no hospitalizations and they saw no deaths from COVID. In the placebo arm, they had 5 people die from COVID during the study. So the Pfizer and Moderna didn't report any patients dying from COVID in either arm. So it's actually, there's more information, I think, about how well Janssen works to prevent hospitalization and death, because there was more severe disease and more disease overall seen in the Janssen study. So just to compare, if you, I think probably several folks have gotten the Shingrix vaccine. That study enrolled 15,000 people, so half to a third of the number of people enrolled in each of these coronavirus vaccine studies, and saw 210 cases. However, it took 3.5 years to do that, whereas it took 4 months or so, 4 or 5 months for each of these studies to enroll and accumulate cases. So yes, it was done more rapidly. It was done more rapidly because we had tremendous support from the community. And so we had enrolled, tens of thousands of people within about 2 months. Each of these studies enrolled all their participants in around 2 months. And then there was so much COVID going on that we got the same kind of numbers of cases that took 3.5 years for the Shingrix vaccine, really in 2 to 3 months for the COVID vaccines. So you can do things quickly if you have lots of people enrolling in studies and you have lots of cases going on. And that's part of it. And the other way you can do things quickly is if the companies don't have to spend a lot of time proving, demonstrating that things work to investors and financiers, they can instead get the data, demonstrate that by all of the measures that we would typically use, that these things work and should be moved forward, and the money was already available. So there were hundreds of people, of companies and industry and academia trying to develop these vaccines. We just haven't seen that with any other infectious disease because so many people wanted to address this. So the other differences between the vaccines are how many doses there are, and most of them are 2. The Janssen vaccine that was tested is a single dose vaccine. The side effects, there are side effects. They're mainly fatigue, headache, sore arm, joint pain, much of which resolves within a day or 2. And we've seen a couple of allergic reactions, but I think I'll have a little bit more on that later. If you think about the millions of people who've gotten the vaccine, the side effects are really no worse than any other vaccine that we've seen. And then that storage temperature again is different between the different vaccines. Okay. This is the data. I think it's nice to see this because it's really impressive, actually. So the red line is people who got the vaccine. The blue line is people who got a saline injection. And then on the graph there, on the y-axis, is the number of people who got sick. And so had symptoms and tested positive for COVID. And so you can see that in the first 2 weeks or so, this is the Pfizer vaccine. In the first 2 weeks, those 2 lines kind of overlap. And then you just don't see many more cases out, to 3 months or so when they stopped it and looked at the data, whereas there's a continuation of increasing number of cases in the placebo arm. The blow up is just those first 21 days, just to show you you start to see that break off around 10 days after the initial vaccine. So this isn't even, this vaccine, you get 2 doses, 1 at day 0, 1 at day 21. You can see that there's starting to be an effect even before you get the second dose. And then the same is true for Moderna. You can see how those 2 curves really differentiate, a little bit before you actually start to see, before the second dose. So the second dose for Moderna was at day 28. You can see that those two numbers actually, or those 2 curves actually spread apart before the second dose. I don't have the data for Janssen because it's not been made publicly available yet, but the curves look similar actually for Janssen as well. Just the numbers are a little different. So that's really the main stuff about the vaccine. There's some special circumstances. They didn't study pregnant women. They didn't study people who had immune suppression from like medication or cancer or advanced HIV disease or things like that. So they did study people who were HIV infected, but they had to be well controlled and have fairly normal immune responses. They did study people who'd had past cancers, but weren't actively on chemotherapy. And the main reason pregnancy is problematic because there are a lot of extra regulations because we don't want to expose pregnant women and their babies to an untested vaccine. And so it's a little controversial, but that's the way the studies were done. There are no safety concerns for either of these circumstances. And the recommendation is if you are pregnant or if you are immune suppressed, that you should get vaccinated if you're otherwise able to, if you're in one of the groups that's getting vaccinated. And people are looking now. There were people who got pregnant. There were women who got pregnant on the Pfizer and the Moderna trials, and there were no adverse events associated with getting a vaccine and then getting pregnant. But there weren't enough to really say for sure how safe it was, but they didn't see any problems with it. We do know for pregnancy that that COVID is much, much more severe. So it's best to protect yourself. Okay. I'm going to skip this actually, unless people have questions. I know people have questions about the variants right now. There are more and more. The two that we've talked about the most are the UK variant and the South African variants. They both have mutations in the spike protein, and the UK one has one in the receptor binding domain. They both currently appear to be prevented by the vaccines, but maybe not quite as well as the original virus strain. The antibodies and convalescent plasma may be somewhat less effective against these new strains. They do appear to be more infectious, and I think are part of the reason why we saw huge sort of spikes in numbers of people and being infected in the UK and in South Africa. They are present in the US and are now being tracked. It's a little unclear, I think, still if they're more dangerous, if they have a higher mortality rate than the original strains. The thought is that these are maybe arising in people who have immune systems that are not effective at preventing, at clearing the infection, and they have sort of long-term shedding and the virus is actually mutated. What we don't know is this going to continue to happen? Are we going to have variants that eventually completely negate the effectiveness of the vaccine? I think those are all things that people are concerned about, but we really don't know yet whether or not that's going to happen. Part of the reason I wanted to tell you a little bit about the immune system is just that that it's not just the antibodies. So the antibodies definitely decrease after you get infected, but we can detect, and that's there. We can detect T cell responses and long-lived B cell responses 6, even 9 months after infection. And we'll be looking at that in the vaccines as well. So even with antibody levels decrease, the immune system has sort of squirreled away information to protect you moving forward. So overall, the vaccines that have gotten EUA are under consideration for it. They were large-scale studies. They have efficacy. They work just as well as most commonly used vaccines. In fact, if we ever had a flu vaccine that worked as well as even the Janssen vaccine, or even some of the vaccines that we think haven't worked very well in this, if we had a flu vaccine that worked that well, we'd be jumping up and down for joy. So they appear to be just as safe. I think people worry about that a little bit in terms of we don't have long-term data, but in reality, for the currently used vaccines, most side effects from vaccines actually happen in the first 2 months after vaccination. So we don't see side effects from vaccines because they're gone, essentially. There's no part of the vaccine left in the body after really a couple of days. And so so we don't typically see side effects that occurred years later. They haven't been studied in people who are pregnant or immune suppressed, but we are getting data on that already. There are a lot of things going on in social media about the COVID vaccines maybe having microchips. It doesn't make sense. They're multi-dose vials. I don't know how you would manage to get the microchip and know which person you injected into if you're taking 5 or 6 or 7 doses out of a single vial. Also, I would think you would see the microchip. We don't make them that small, and if people really wanted to track you, they would probably use your phone anyways. There's also a social media thing that says that they cause infertility, and it doesn't make any sense at all. So, and there's no data, and clearly people have gotten pregnant after they've gotten the vaccine. So anyways, I'm going to end the stop the sharing, and then we can answer questions, or however you want to do this, George.
Thank you, Rachel. That's great. I know you have already answered some of the questions
Yeah.
that came in. If you could go through, whichever you feel like answering, please go ahead, and that might enlighten us a little bit more, and then people can chime in and ask questions as well.
Yeah. So, okay. So one of the first questions, and I think this is, infection rates and deaths seem to be going down. Are we doing something right? Well, hopefully. What does the spring look like? I wish I had a better crystal ball. So there are a couple things going on. I think we have been doing something right. I think that it's clear that more and more people are wearing masks. There definitely have been, the uptake for vaccines has been, it's been problematic, I think, getting it rolled out in a reasonable way. And part of that has to do with the fact that the storage is complicated and the distribution is complicated. Um, the other thing to think about is that coronaviruses are winter viruses, and so I think the winter spike that we've been getting could also be in part predicted by the fact that this is the season where you see coronavirus infections. And so I think the combination of people getting vaccinated and the weather getting warmer should make the spring better. I think moving on to the second question and sort of taking it from there, the variants could make that a little bit more complicated. So what we don't know is if the variants took off and spread, would that cause a spike like they saw in the UK? So I think that's the reason why the public health messaging, I think, sometimes feels a little confusing, because we say get the vaccine is our way out of this. But then once you get the vaccine, we say you still have to wear a mask and you still have to socially distance and still do all the same things. I think, frankly, until we get better control of this, we should do everything that we know works. And then when we see the numbers continue to go down, and when we see what happens with the variants, that we can start to open things back up. And if you get the vaccine, it's clear that even the South Africa variant still provides, there's still protection from that against severe disease and death. So the vaccine really prevents you from getting really sick, which is what we really need to get past this. I don't think we're going to make the coronavirus go away. I don't think COVID's going to go away, period. But I do think we can get to a point where it becomes more like the flu or the seasonal coronaviruses, where if we can protect the people who are really, really at high risk by vaccinating them, that we can start to be able to treat this. I think also it's clear that the hospitals have gotten better at treating COVID, and that if they're not overwhelmed, then the mortality rate goes down. There's a question about why the vaccine rollout is being done so poorly. We don't have a good answer for that, except that I think it is really complicated, and we have not funded our public health system over the past years and decades the way we probably should. I think to some extent the public health system is the victim of its own success. So we had a better public health system back decades ago, when there was still a lot of syphilis and tuberculosis and other infectious diseases that we needed to deal with. As those infection rates went down, we figured, okay, we're fine. We can start cutting the public health infrastructure. And I think it's clear that we need to be better ready as a country to deal with these kind of pandemics. I do think there's infrastructure that that was available to do, for example, to do the vaccine studies. There was pre-existing infrastructure, and that's partly why that actually worked really, really well. But there wasn't as much pre-existing infrastructure for distribution of vaccines on a public health measure, and that's why that has not gone as well. So it's a matter of sort of funding and priorities and so on. So there's a question about long-term effects of COVID. I think people are still trying to understand that there are post-infectious long-term effects of a lot of infectious diseases that are, seem to be sort of lingering side effects. I think we're still trying to figure out what those are and what's the best way to treat them. It's not clear that they're infectious, that they happen long time after the virus is gone. And, we have difficulty, I think, sometimes in then treating something where we don't understand how it works. What I've told people, there are some clinics that are in the community that are following what they call long haulers, people who have chronic symptoms months after they've gotten over COVID. Some of it is if you've got very, very severe COVID and you were in the ICU for a month, doesn't matter what disease you get. If you're in the ICU for a month, it's probably going to take you, a year or more to recover from that. It's just a sort of debilitating both to your body and to your brain. And, what we tell people is to do the same kinds of things that we tell people to stay healthy in general. Make sure you're getting enough sleep, make sure you're getting exercise to the extent you can, make sure that you're eating healthy. And unfortunately, we don't have a whole lot more than that, although it's always reasonable to see somebody in one of these long hauler clinics. So when will life return to normal? I don't know what normal is going to be. I think it's likely that schools will return in a much more normal sort of way in the fall. To what extent are we going to need to continue to wear masks? I don't know. I think eventually we can maybe stop wearing masks some of the time. I will tell you that we have had like nothing of a flu season this year. So masks actually work for other respiratory viruses very, very well. And so we may not have completely knocked COVID, but we did knock the flu by wearing masks. So I think there is some thought that maybe it's not an unreasonable thing to wear a mask in the winter virus seasons, when you're in large groups, in large crowds, and so on. Best information for the disease. So there are a lot of good websites. So CDC puts out good information. Johns Hopkins has a great dashboard that gives you information about the sort of progress and both vaccination and infection. And then actually the New York Times has some really nice stuff as well. So those are the three places that I look when I want like a thing. And actually, I think both the city and the county health departments now have decent websites that tell you how to get vaccinated, how to, what's going on with the disease and get good information. So what surprised me the most? I guess what surprised me the most is how political it got. It seems like it shouldn't have gotten political. It seemed like we were all sort of should have been fighting the same virus with the same sort of scientific information. And I think that's what surprised me the most. Anyways, okay, I'll let other people talk. I'm sorry.
Okay, so You can raise your hand or just ask question. I think maybe you can hear. I see, yeah, I don't see anything in the chats.
I have a question, George. Is this going to be like the flu where we'll need to get some kind of a shot every year? And if so, like when would that start? So for example, I'm getting my second shot this week. When would I need to get another one? Or have they even looked at that yet?
I think we don't know. I don't think this is going to be like the flu. Coronaviruses don't change the same way the flu does. So the flu actually changes every, the way it replicates, it changes its surface proteins every year. And so we know that the vaccine doesn't work for the next year. You need to keep updating it. So my expectation is that we may need to cover the variants. And we may figure out, because this has happened with other vaccines, we may figure out we need a booster, and hopefully it would be a booster in a year. It does look like immunity, I showed that the immunity starts actually fairly early, but it does appear that immunity continues to improve actually after the vaccine, for the first 3 months, 6 months or so. Part of it is we just don't have that information yet. So I don't think we can say for sure. I don't think this is going to be another shot that you get annually, like the flu vaccine. But I think we may need a booster. We may need to cover variants as they come up.
Thank you.
Oh, it looks like there's something about other countries in Europe doing with vaccines. They've not done as well even as we have. I think it's improving. So it's, infrastructure for doing these and getting this done has been highly variable. Getting access to the vaccines has been somewhat variable. I think there is a concern that we're going to wind up, that rich countries are buying up the vaccines. And I really do think the hope was that that Janssen and AstraZeneca would be effective. There's some concern about AstraZeneca not being as effective against the UK strain, not UK, against the South African strain. And so South Africa had a ton of AstraZeneca vaccine to give their folks and then pulled back on it. So both of those vaccine platforms and those companies have given like the infrastructure to give the vaccine throughout Africa, because they were using them for the Ebola vaccination process.
Right. So there's a question from Nigel. What's the best arguments against vaccine skeptics?
Yeah, I think it's mainly just talking to them and finding out what their concerns are. So what I've heard mainly from people, so I mean, if you have somebody who is just a rabid anti-vaccine kind of person, I think that's going to be hard. If they've never gotten a flu vaccine, you're probably not going to talk them into a COVID vaccine. If they, really do think that they're going to be, that there's going to be a tracker that the government is going to track them with, if they get a COVID vaccine, if they don't, if you can't get past that, the main thing that I've seen is that people just don't, they think it was done too fast and they think it was political. And I think just the information, that these were done with more people than other vaccine studies that are, for commonly given vaccines like Shingrix and hepatitis vaccines and the Pneumovax and, all these vaccines that we give people all the time. If you let people know, like, no, this was not done in a smaller group. This was done with more people, and there were more, there's more data about how well they work. Also, what I've heard from a lot of people is, well, I'm not going to be the first one to get the vaccine. And generally what I tell them is, oh, you are far from the first. Like, this has been in, you know, millions of people at this point, and we just want to make sure that you have access and that you're in line. So other than being on several lists, do I have any websites where you could sign up for vaccines? I don't have good, I think you still have to find the different vaccine, you know, sign up with your, you know, if you're Mercy or BJC or SSM, they all have sort of lists that you can sign up on. The county and the city have lists as well. You know, I think that is part of the problem. Hopefully, eventually, this is going to wind up being in you know, your local Walgreens and be easier to get, but it's going to take a while before it gets to that point. There's a question about retransmission by vaccinated individuals. Yeah. So so it is clear that vaccine doesn't prevent you from getting infected. It prevents you from getting sick. And so there is concern that vaccinated people could still get sick and not sick, could still get infected, could still shed the virus and potentially pass it on to other people. And that's why we tell people after they get vaccinated to still wear a mask. So the masks have never been to protect, they're a little bit to protect you. The masks are mainly to protect everybody around you. So it's a public health thing, because the masks work very well to prevent you from transmitting to other people. They work slightly less well at you getting the virus from somebody else who's not wearing a mask. So if everyone wears a mask, then you get that double protection and people are most protected in that situation. And then there's a question about why didn't this virus disappear and when SARS and MERS did. Some of that had to do with the fact that SARS and MERS were, well, two things. One, they were much more virulent than COVID. So they had a higher mortality rate. And we tend to see that if something kills people too quickly, it often kills people too quickly for them to pass it along to too many other people. So there's that. But then the other thing is, it is pretty clear that people are infectious. Their like peak of infection risk for transmitting COVID occurs just before they're symptomatic. Whereas SARS and MERS, you weren't really transmitting until you got really sick. And so then you can tell people, if you've got a fever, if you feel sick, stay home. Whereas you are probably, if you've gotten COVID, you may be transmitting to people for several days before you even know you have COVID. And so I think that's what was happening, was it just made it much harder to actually control it. And now it's in so much of the population, it's likely to become like one of the seasonal coronaviruses where we'll always have it, but hopefully less virulent, less of a public health problem. If you've had COVID, you should continue to wear a mask. It's clear that if you've had COVID, that you are somewhat protected from getting COVID again, but not completely. So you can get infected again. You could get infected the same way as people who've been vaccinated. You could get infected and not have symptoms and then transmit. So you should be wearing a mask so that you can protect other people. So.
Anyone else have a question? Any hands up, or I don't see anything more in chat. Tim, your question was answered. Tim had hands up.
Yes, it was.
Okay.
So Rachel, I just put it in the chat, but maybe you could talk a little bit more about the recent CDC advice for double masking.
Yeah. So I think we should be practical with this. So double masking clearly works better. A lot of it depends on what kind of mask you're using. So nobody should be double masking an N95. That is horribly uncomfortable and unnecessary. The N95s actually, it has to do with sort of layer of filters. So N95s actually have multiple layers in them. And some of the surgical masks that they use in the hospital also have multiple layers in that single mask. So the issue really comes up with if you've bought the surgical type masks, online, and you're maybe not as certain about the quality of it. The double masking, what seems to work best, I think, about it is that it fits better. So the mask should cover your nose and your mouth, and you shouldn't have air coming up the top or coming out the sides, because then it's not doing any good. It's still going. The idea is you don't want to breathe the same air that other people are breathing. And so you want to be breathing through the mask. So what happens is if you use a surgical kind of mask, but it has gaps on the side, and then you use a cloth mask that fits better over it, you're more likely to have it kind of tailored to your face in a way that that protects you better. That being said, if the mask is making you so uncomfortable that you can't breathe, or that you have to keep taking it down to talk to people, or to feel comfortable, then that's not doing you any good at all if you have to take it down. So if I think what some people have said, and the other thing is people say that it hurts their ears, which I totally understand. What you can do, there are the masks that tie behind your head. Sometimes I think people find those more comfortable and find it easier to get those to fit in a way that actually covers your mouth and your nose. So yeah, I think if you are mainly staying at home, and you're going out to a place like a grocery store, you're going out someplace where you're worried, and it's a limited time frame, and you can do it comfortably without feeling like you're suffocating, then it's probably a good idea to double mask.
And then for us sort of lay people, do you sort of consider the KN95s to be almost equivalent to an N95 for a lay person?
Yeah. I mean, they're not exactly the same, but they're probably, and it's going to be uncomfortable to wear something in addition to a KN95. Right. Yeah, if it fits to your face and you don't feel, if you don't feel air coming up, up to your eyes and out to your cheeks, so if you breathe in and out, and you can feel the mask moving in and out and not air going out the sides, then it's fitting well, and it's probably effective.
What about people like Brian and me who have beards?
Yeah, so the N95s don't work very well when people have beards, because it does sort of leak out. And so then you might want to think about, if you can get it to kind of cover, or if those are the situations where, like, the masks that have a tie sometimes will fit better.
Okay,
so.
Thanks.
I think we are at 5 o'clock, but Rachel, before we go, I'd like to, there was one question that came in from Trinity that I'd like to ask, which is really about the healthcare workers. And those are, you guys were are for the last 1 year have been doing the heavy lifting. And the question was, how can we be advocates, or how can we support? What can be done?
Yeah, I think what hurt healthcare workers the most was people not wearing masks and not taking things seriously. Because you feel like, and people saying things like it's just a cold, that we're overreacting, or we're doing something, when, if you've been in the ICU and you've seen people dying, then it's sort of an extra sort of pain to have people say this is not that big of a deal. And so one of the things that really upset me always was that people are like, it's just like the flu, and I'm like, well, the flu is actually pretty bad too. We see people die from the flu as well. So that kind of stuff, but otherwise, I feel like healthcare workers have actually gotten a lot of support. And, the people that I feel like we should be supporting are, the grocery store workers and the delivery drivers, and the folks in sort of, EMT and police and stuff, you know, who still are being, even the people working in restaurants. So I think, you know, be kind to other people, recognize that everyone's sort of dealing with this their own way, and then be patient. I think, you know, we all want to get back to something normal. And I think we've gotten a lot of love and from the community, and I really do appreciate it. So.
Well, thank you very much, Rachel. This has been wonderful. I really appreciate within it in your busy schedule to, you know, that we were able to find this 1 hour to have your time with us. So
Oh, no.
really appreciate it. Thank you very much.
Thank
Thanks,
you, Rachel.
everyone.
Thank you all for
Thanks,
joining in today.
Rachel.
It's nice to see everybody.